I'm Zach Hines, and it's my great pleasure to introduce our next speaker, Professor Stephan Herzig. So Stephan is a department head at the Helmholtz Diabetes Center and also the director of the Institute for Diabetes and Cancer at the Helmholtz Munich. In addition to that, he is a full professor of molecular metabolic control at the Technical University of Munich and an honorary professor at Heidelberg University. Since 2018, Stephan is the editor-in-chief at Molecular Metabolism. So if you have any exciting results, you know who to see after this talk. He's garnered multiple awards and recognition for his work over the years. Most recently was the Camillo Golgi Award at the EFSD in 2023. I think it's been quite interesting that he's made several unique advances both in the fields of cancer and in diabetes, underlying these elements of metabolism as an interlinking mechanism. And throughout his career, he's uncovered several insights into how breakdown in fat and liver metabolism underlie diabetes pathogenesis, with the ultimate hope of informing and identifying new treatment strategies. So with that, please, let's welcome Stephan to the stage.
Zach, thank you so much. Thanks for the invite. It's really an honor to be here. This is also my first time in Copenhagen this week, which tells you that it's always great to come back. And this is really an impressive audience, so I'm really happy to be here. I realized that my pictures look identical to my picture, which is good. So I haven't changed that much over the years, obviously, which is good. I wanted to talk and touch upon something that we've been working on for the last, 2 decades, I would say, which is fasting metabolism. And why is that? And I think this cartoon just highlights when you work in metabolism research, then maybe the most critical endocrine dysfunction that you can experience is insulin resistance. So the inefficiency and inability of insulin to work properly does not only affect, of course, one particular organ system, but it has really multiple impacts on many different organ compartments. It leads to lipid problems, leads to hyperglycemia, leads to, as a component of obesity, can go all the way up to cancer, cachexia, and hypertension. So, insulin has to work properly, of course, in order to avoid metabolic complications. So we have, in many studies over the past years, worked in the liver and on liver metabolism. And of course, we know these days quite well what insulin actually does in the liver and obviously also in other organs. So in times of refeeding, you build up energy blocks and you store energy basically to then have that available whenever you starve. And so we became really interested on the left part here on the fasting side of things in the liver. So because there are obviously many pathways that are then necessary to survive starvation periods, and many processes are going on that then are actually prime the body to survive times when you sleep or when you have not seen food for a long time. And in the condition of insulin resistance, of course, there's at least a relative overactivation of some of these pathways that then can drive certain pathologies, including, for example, hyperglycemia, as we see it in diabetes. And it turned out, so working on fasting became really popular, in particular in the very recent times and in the lay public, because it turned out that intermittent fasting, so voluntary times of not eating, are actually quite beneficial. And this has been shown in many studies in mice and also in humans in clinical studies. Whenever you do intermittent fasting, this has quite beneficial effects on lowering glucose, lowering lipids, you improve your blood pressure, and so forth. And indeed, in a clinical study that we ran in our Heidelberg lab together with the colleagues there, it turned out that patients with diabetic nephropathy could actually improve their kidney function once they were on this intermittent fasting protocol. And not only that, along with this intermittent fasting, maybe as predicted, we heard about ketones just a minute before. This fasting paradigm increased ketone bodies, and it also improved the HOMA-IR. So telling us that in general, obviously, maybe we can employ fasting pathways and fasting mediators to improve metabolism under certain conditions, as just exemplified by this little study here, as I said, that we ran in patients with diabetic nephropathy. So I want to do, in the next few minutes, I want to touch upon three aspects basically of fasting. One is rather short, and this is going back to some molecular analysis. How does fasting work, and what are the mediators of fasting metabolism? And all of this started actually 20 years ago when I was a postdoc at the Salk Institute with Mark Montminy, and we discovered that there's one particular transcription factor, PGC1, many of you will know this one, actually coordinates the interaction between two main endocrine pathways in the control of fasting metabolism. One is the cyclic AMP pathway, mediating the activation of the transcription factor CREB, which induces the transcription of PGC, and the other one are glucocorticoid hormones, which are also elevated during fasting. And then PGC turn out to interact with the glucocorticoid receptor and thereby synergizes basically and mediates the synergy between these two fasting pathways, driving gluconeogenesis essentially under conditions of insulin resistance and in diabetes. Now, we have continued, of course, to identify checkpoints, molecular checkpoints in fasting metabolism over the years. And most recently, I just give you a very short example of one of the things I think where many things still have to be discovered. We became interested in what actually modulates the activity of transcription factors that might drive certain fasting aspects. And we screen for post-translational modifications that would be altered in response to fasting. And so here's one example where we looked at sumorylation. So it's a small 11 kilodalton peptide that is attached to proteins and then can alter its activity and function in multiple ways. And so we did a screen and fasted and refed animals and then asked the question, do we see differential sumorylation of proteins? And as it turned out, there was one transcription factor, which is called PROX1, where is it? PROX1, that turned out to be specifically sumorylated in response to fasting and feeding. In fact, what happened, and this is shown in the next slide here, upon fasting, we would lose the sumorylation of this transcription factor, and in refeeding, the sumorylation was there. So it was obviously, it's a post-translational modification of a transcription factor in response to nutrient availability. And the important thing is, I don't show the data. If you would look at the same picture in animals fed a high-fat diet, so in pre-diabetes, glucose intolerant, obese, this regulation did not occur. Basically, the sumo signal and the sumo peptide stayed attached to the PROX1 transcription factor under these metabolic dysfunction conditions. So obviously, this switching back and forth of sumorylation was something that was really relying on the physiology of fasting feeding cycles. And then, of course, the question was, what does it do? Is it meaningful by any way that we have the sumo attached to this transcription factor in response to fasting and feeding? And well, the short answer is we generated a knock-in mouse that replaced the one sumo site on PROX1 and then fed the animals high-fat diet and then asked the question, what is the phenotype? And I can tell you there was just one single phenotype related to this inability to sumolate or to desumolate this transcription factor, and that was cholesterol metabolism. It was a little bit lower circulating levels of LDL and HDL cholesterol, and that was the only phenotype. And I'm just using this example, and this, I should say, and this partially overlaps with a complete loss of function of PROX1 in the liver, where you also have an impact on cholesterol metabolism. And we found that this specific sumolation, obviously it was in control of the bile acid detoxification pathway, which then in turn controlled this cholesterol metabolism. So I'm just using this one little example without going into details to say, well, there are probably way more things that we have to learn in terms of basic molecular biology of how fasting actually regulates relevant pathways under these conditions. And the sumolation is just one example that is nutrient dependent and that can control very specific aspects of fasting metabolism. So as a summary, basically over the past, as I said, 20 years, we have identified a number of transcriptional checkpoints. I talked about the PGC1 already. There are other cofactors, mostly nuclear receptor cofactors that control this fasting feeding cycle in the liver, and that they then have not only an effect on the intrahepatic regulation of metabolism, but also on systemic aspects, including the control of insulin sensitivity, bile acid homeostasis, cholesterol, triglycerides, and also, of course, glucose. And we think this is quite exciting that something within the liver can control the systemic aspects, because in the long run, this will, of course, have an effect then also on the long-term complications, for example, of diabetes, if something goes wrong here at the molecular level. When we had this picture and had been working on this for quite a number of years, 20 years or so, as I said, there was one thing that we had not done. And we had never looked at the interaction between different cell types in the liver and asked the question, how important is now the hepatocyte to drive all these systemic effects, or is it communicating with other cell types in the liver? And that was the time when Anne Loft, who is now back in Odense, joined the lab as a postdoc, and she, together with Anna Alfaro, initiated a study where we tried to identify or address the question, how do the hepatocytes communicate during fasting with other cell types in the liver, and is that important for fasting metabolism in general? In order to address this, we generated 2 mouse models. Essentially, in the one mouse, we tagged the nuclei of hepatocytes, specifically with the GFP, and this works quite well because you have in a cell selective manner if you combine this then with an appropriate Cree driver. And then the second mouse model is doing the same thing, just labeling the nuclei of macrophages. And this also worked relatively well in terms of cell selectivity. So basically, we could now take these tagged cells, isolated from the liver, and employ fasting regimens to ask the question, what is the response to fasting, not only in the hepatocytes, but also in the macrophages, so in these inflammatory cells. And then we can run, of course, all kinds of omics studies. Now, if we did this, there was one not so surprising thing and one maybe more surprising thing. The not so surprising thing was that in the hepatocytes, of course, there is a difference between fasting and feeding. So the hepatocytes respond in their transcriptome, of course, to fasting and feeding cues, as we would have expected and we know for many years. What was maybe more surprising is that also the macrophages, there was a distinction between the transcriptome and the fasted and the refed state, in indicating that it's not only hepatocytes, but it's also the macrophages in the liver that seem to respond to fasting. And then, of course, you can ask the question, okay, what does it mean? How is it? What does happen? And who's controlling this whole fasting response in macrophages? Now, then you can take all the omics data, of course, do motive analysis, and then find a list of transcription factors, again, that are regulated by fasting signals specifically in macrophages. And well, you can find maybe a few suspects if you talk about macrophages. The STAT2 activity was changed. Then we have some inflammatory transcription factors that are changed. But then we had this one here that showed quite a robust upregulation in response to fasting. And this is a nuclear receptor, NR3C1. And behind that is the glucocorticoid receptor, which was, of course, quite interesting because glucocorticoid, I will allude to that in a second, have been studied quite extensively in hepatocytes already and are known to be important for driving fasting responses in hepatocytes. Now it turned out that also in the macrophages, in response to fasting, this important fasting-induced pathway, endocrine pathway, glucocorticoid, seemed to be activated as demonstrated here by the activation of the glucocorticoid receptor. And then, of course, the question was, does it have any impact on fasting? So we generated a mouse. Actually, we generated 2 mice, but this is just data from one, that lacked the glucocorticoid receptor specifically in macrophages, and then fasted these animals. And so on, the one phenotype that we observed was this one here. These mice failed to mount a full ketogenic response. In other words, if the glucocorticoid receptor was missing only in macrophages, the liver could not generate the full amount of ketone bodies that it would normally generate in response to fasting. And this was really also very specific because no other main metabolic pathway was affected by this manipulation. And I just summarize of what it turned out to be in this little cartoon here. So it seems that in response to fasting, also the macrophages in the liver respond to glucocorticoid hormones. The glucocorticoid receptor actually controls a distinct set of secreted factors that then travel to the hepatocyte and drive in turn a specific subset of ketogenic genes controlled by the glucocorticoid receptor in hepatocytes and the nuclear receptor PPAR alpha. And by this, you can then mount the full ketogenic response. So telling us that indeed the full fasting response by the liver requires not only the activity and the action in hepatocytes, but does also require the intrahepatic communication between macrophages in this context and hepatocytes on the other hand. And this was quite intriguing for us because, as I said, we had worked on fasting endocrine pathways for quite some years before, and so far only concentrated on hepatocytes. So this was the first time that we saw that also glucocorticoid receptor signaling was important in a non-parenchymal cell type in response to fasting. And it was placed quite nicely into the previous work where we had identified many of the signaling pathways and metabolic pathways that are actually under control of the glucocorticoid receptor in the liver. I talked about PGC already. Then we have regulation of triglyceride metabolism by the glucocorticoid receptor by controlling this transcriptional repressor, which is called HES1. The glucocorticoid receptor also controls hepatic bile acid metabolism, and it also controls protein metabolism. So all of this together then gave a very nice picture, having identified multiple pathways that are under control by glucocorticoid hormones in response to fasting in the liver, both in this case here in hepatocytes, and as I showed you in the previous example, obviously also controlling at the same time the interaction between the hepatocytes and the non-parenchymal cells in order to mount a full fasting response by the liver. Now, all these data together then show that, of course, obviously this endocrine pathway has quite an impact on fasting metabolism in general in the liver. But all of these factors that we had identified at that time were, of course, proteins. So we had PGC as a transcription factor, HES1 as a transcription factor, we had a transporter, NTCP, and so forth, all under control of the glucocorticoid hormonal pathway. At that time, we then asked the question, well, if glucocorticoid hormones are so important and seem to mediate many of the fasting aspects in the liver, can we actually identify a new layer of glucocorticoid hormone-dependent metabolic control? And that was the time when we turned our attention to microRNAs, so small non-coding RNAs, and we asked the question, can we actually identify glucocorticoid-dependently regulated microRNAs in the liver that would also be important in metabolic control and maybe fine-tune some of these aspects that we had looked at before. So we screened a number of we screened microRNAs, basically asking the question, which microRNAs in the liver are regulated by glucocorticoid hormones and which at the same time are dysregulated in diabetes and obesity. And I make a long story short, it turned out there was a bunch of microRNAs, and this is actually the second topic that I want to talk about, because it turned out that these microRNAs that we originally identified to be under control of the glucocorticoid pathway might have some therapeutic benefit if we manipulate them in the right way. This cartoon summarizes our initial finding that we had. We identified one specific microRNA, microRNA 379, and this part of a cluster of microRNAs. It's actually a conserved cluster between mice and humans. As I said before, it's under control of glucocorticoid hormones, but probably also by other endocrine pathways. And what happens then if you activate these microRNAs, this microRNA here, you will downregulate specific lipoprotein receptors in the liver, and that leads to an inhibition of lipoprotein and triglyceride and cholesterol uptake into the liver. In turn, if you inhibit, and that's now the point, if you inhibit this particular microRNA here, 379, you can achieve the opposite. You actually induce the expression of these lipoprotein receptors. You enhance the uptake of LDL and VLDL particles, and you have quite a substantial lowering of triglyceridemia and cholesterol levels in the circulation. And that's when the idea was born, maybe we can use inhibitors of these microRNAs to actually do something beneficial under conditions of metabolic dysfunction. We had seen that with this particular microRNA, we were able to substantially lower hypertriglyceridemia in these animals. We then went into the clinics and to our clinical departments, looked into the biobanks there and asked the question, beyond 379, do we have other microRNAs from the same cluster that might also be deregulated under conditions of obesity and diabetes? And that turned out to be the case, and I'll just give you one example. It turned out to be microRNA 541. You see 379 on top, 541 here. And these are liver samples from patients and with obesity and diabetes. And you can see that these two microRNAs are quite substantially upregulated in obese and diabetic patients. And they correlate with certain clinical parameters, including triglycerides and also HOMA-IR, which gave the idea that, well, obviously beyond all the proteins that the glucocorticoid receptor regulates, it seems to regulate also microRNAs in this particular cluster, and there's a deregulation of this pathway under conditions of obesity and diabetes, not only in humans, as I showed you here, but you can actually see the same thing in many different animal models of obesity and diabetes. So, as I said before, the initial idea was, can we use this somehow therapeutically? Because we, as I said before, with an inhibitor specific to 379, we were able to lower hypertriglyceridemia quite substantially. So what we did is we designed an inhibitor molecule that would be able to target either one microRNA specifically, 379, or inhibit 541, or in a combinatorial way inhibit these two microRNAs at the same time, and we could do it in a way that this was achieved in a liver selective, even hepatocyte selective manner. And then what happened is shown below as one example. This is a glucose tolerance test. Inhibiting 379 alone gave some improvement in glucose tolerance. Inhibiting 541 was already a little bit better, but then actually when we inhibited both, gave quite a substantial and remarkable improvement in glucose tolerance by delivering this specific inhibitor compound into the hepatocytes. Now, what was the other phenotype if we employed this inhibitor molecule? As I showed before, quite substantial improvement in glucose tolerance. Here you see also insulin tolerance was improved. This is a pyruvate tolerance test, so it blocks also gluconeogenesis in the liver and basically doubles insulin sensitivity in these models. So quite a remarkable effect on metabolism by inhibiting these two small microRNAs at the same time synergistically. Question was, does it also, these were actually lean and chow-fed animals, does it also work in disease models of obesity and diabetes? This is high-fat diet, and you can see that it lowers HOMA-IR quite substantially. It improves triglycerides, and it improves also cholesterol and LDL. So a very broad improvement in metabolic parameters upon the inhibition of these two microRNAs in a synergistic way. Now, these were type 2 diabetes models, and then the question was, since we had seen also upregulation of these microRNAs in type 1 diabetes, so what about type 1 diabetes? So we used our inhibitors in a model of type 1 diabetes, STZ injection. This is the glucose curve, as you would expect. Quite high levels upon STZ injection, and this actually then happened when we gave our inhibitor. So you basically normalized the glucose phenotype and the glucose tolerance and insulin and glucose tolerance in these type 1 diabetic animals without touching the pancreas, I should say. So this is all done by peripheral mechanisms, probably mediated by liver function. So I summarize this part with this little cartoon here. We think that we have identified a cluster of microRNAs, and we can synergistically inhibit those microRNAs, and they then have a broad beneficial impact on metabolism. They lower glucose, improve glucose tolerance, insulin sensitivity, cholesterol, and LDL. If we delivered this specifically to the hepatocytes, and the big thing is, it's all under weight neutral conditions. So there's no loss in body weight, which is different, of course, to the current incretin therapies that I think we're going to hear about later in the day today. So all the improvements are really meant and achieved by this hepatocyte-specific action, irrespective of any weight loss. And we do think that there are still patient groups who might actually benefit from this if we were able to bring our inhibitor to the right place at the same time. Okay, that was beyond the first part, basic aspects of fasting. Here, this was the idea on how we may employ fasting regulated mediators as new, maybe therapeutic approaches to counteract metabolic dysfunction. And the final part is going to be looking at an aspect of fasting, so to speak, that covers a little bit of involuntary fasting or wasting, a condition that we became quite interested in, which is cancer cachexia. So it seems to be completely opposite to obesity and diabetes, but in fact it's not. I should say that cancer cachexia, so the involuntary loss of body weight, muscle mass, adipose tissue mass, is actually associated with a number of features that you might also see in obesity, despite the obviously different appearance. But you do have a certain degree of insulin resistance, you do have inflammation, etc. And this is the important thing, the majority of cancer patients is actually affected by this involuntary wasting condition. In pancreatic cancer, for example, 80% of patients are affected by cachexia. So they are not able to maintain their body weight. And even if you gave them extra calories, it does not help. Once you're on the trajectory to weight loss, it's gonna go down no matter what. And it's a huge unmet clinical need. And there's actually not a single FDA-approved drug so far to counteract cancer-driven cachexia. So we became interested in that because we felt there are many similarities in metabolic control between obesity and diabetes on the one hand, fasting metabolism on the other hand, and then this cancer-driven body weight loss. And in fact, it turned out very recently, this was a study led by Maria Rome in our institute, who discovered in a cohort of patients that who developed cancer eventually, that patients who had diabetes before the cancer diagnosis actually developed more severe cachexia as compared to patients who were not diabetic before their cancer development. So in other words, diabetes actually reduced their survival and also increased weight loss and increased inflammation in these cancer patients, and overall indeed aggravated the cachectic phenotype. So these days we would consider maybe even not only cancer by itself, but also cancer cachexia as a long-term complication of diabetes. As I said, we are liver people, and classically in the field of cancer cachexia, most people would look into skeletal muscle and adipose tissue, which is of course reasonable because that's what's lost. But there were not that many studies really asking the question, what happens actually in the liver? And is the liver at all important for the cancer cachectic phenotype, so for this involuntary wasting syndrome that is associated with cancer? So basically, and that's the final part, we then probed what is the hepatic response to cancer cachexia using different set of mouse models, including mouse models where we implanted tumor cells that then develop cachexia. We have controls that have a tumor but do not develop cachexia. We have, of course, PBS, all kinds of and different types of tumors, including colon cancer cell lines, pancreatic cancer cell lines, and so forth. So we took all these animals and then basically employed our mouse model again that we used for the fasting study, where we could pull out the isolated nuclei from hepatocytes from all of these animals, asking the question, what is the hepatic and the hepatocyte response to cachexia in this setting. And just to show you that these models actually work, so the pro-cachectic tumor cells, they actually cause weight reduction, whereas the controls don't. And then, as before, as I said, we isolated the hepatocytes and then ran high throughput analysis to ask and see what actually cachexia would do to the liver. And as it turned out, there are indeed very distinct transcriptomic responses in hepatocytes under cachectic conditions. Indeed, the controls all quite nicely cluster both in the whole liver but also in the hepatocytes. And then the cachectic animals are cluster also as well, and these are the two distinct tumor types that are in different mouse backgrounds. But overall, the first conclusion was, yes, indeed, the hepatocytes respond in terms of transcriptomic answers, so to say, under cachectic conditions. And then, of course, you do the analysis, and it turned out that there was one cluster of genes in the hepatocytes. This was this cluster GC3 in particular, where there was an upregulation of genes, in particular in the fully cachectic animals in this C26 colon cancer mouse model. And then as it turned out, and you can do the analysis, what kind of transcription factors actually control the genes in this cluster, and then it turned out, well, we have a few usual suspects. STAT3 signaling, some NF-kappa B related signaling, but then motive changed in particular for these nuclear receptors here, NR1, D2, and D1, and also ROR. Now, and these ones might be familiar to the people who actually work in the field of the molecular clock, because these are key components of the circadian rhythm responsible clock. So, because NR1D1 is actually a reverb, and the RRs are sitting here in the stabilizing loop of this of the molecular clock that is, of course, driven mostly by clock and BMAL. So the conclusion was that under cancer cachectic conditions in the liver, there's obviously some kind of disruption of this molecular clock, as evident by the dysregulation of the motives of these transcription factors that are part of the stabilizing loop, in particular the reverbs. And indeed, it turned out, this is the rhythmicity of gene expression of the reverbs here in healthy mice. And then if you compare this to the tumor-carrying animals, there was indeed a major downregulation of the reverbs under these cancer cachectic conditions. And so the question was, of course, does that have any functional meaning? So what we did is basically we restored the expression of reverb, in particular in the liver of cachectic animals, by delivering a construct, an overexpression construct with an AAV into these animals. And the phenotype was quite remarkable, I have to say, because as you would have predicted, in control animals, cancer cachexia then leads to the loss of body weight quite substantially. If we restored reverb specifically in hepatocytes, this prevented, to a major degree, body weight loss. It actually increased then heart weight, it increased skeletal muscle mass, and adipose tissue mass. Did not have an effect on the tumor, which is in a way a good control, that it's not just regulating tumor mass and thereby influencing cachexia. So as a conclusion, this cartoon here turned out that in the liver, obviously, there's a distinct transcriptional response to this involuntary wasting and fasting in cancer. And there's a disruption of the circadian clock, as demonstrated by the dysregulation of reverb transcription factors. And data I have not shown you, but as it turned out, reverb seems to control, again, a distinct signature of secreted factors that are traveling actually from the hepatocytes then to peripheral organs, including the heart, including skeletal muscle, including adipose tissue, and mediate the cachectic phenotypes in terms of lipolysis, muscle wasting, and also heart atrophy. With this, I'd like to summarize that Obviously, fasting is important for healthy metabolism. We do think that fasting metabolism, in particular in the liver, is not only controlled by things that go on in hepatocytes, but you need intrahepatic communication between different cell types in the liver to mount a full and proper fasting response. We do think that, as an example here, these microRNAs that are under control of fasting-driven endocrine circuits might provide the avenue towards new therapies with their beneficial effects on many metabolic parameters under weight-neutral conditions. And finally, we do think that also hepatokines steer systemic metabolism under involuntary wasting conditions, as we, for example, see it under cancer cachectic situations. With this, I'd like to thank the group. I mentioned Anna already, who ran the first study. Sören Fisker-Smith, who is also back in Odense, ran predominantly the cancer cachexia study. I'd like to thank all collaborators and funding agencies, of course. Zach was so kind to mention, if you do have good stories, feel free to send it to Molecular Metabolism. If you send it today, you have the rejection tomorrow, I promise. We're really quick. And finally, then, you're, of course, all welcome and invited to join now the 11th Helmholtz Diabetes Conference, taking place, as always, during Oktoberfest times in September this year in Munich again, with the focus on wasting metabolism. Thank you so much.
Thank you very much for a really exciting talk. I know I have questions, but I think maybe we'll let, yeah.
Hey, thank for a great talk. So my question is about ComboMir. You have mentioned that ComboMir is regulating LDL receptors. Am I right? Yeah. And this regulates the LDL receptors, and inhibiting that miRNAs would upregulate LDL receptors in the liver and increase the uptake of the fat in the liver. So have you checked the fatty liver conditions of these animals before?
Yep. Does not happen because it does a few additional things that I try to hide in the cartoon because I can't tell you yet. But as it turns out, it obviously at the same time stimulates also mitochondrial biogenesis and connectivity and activity. So we do think that all the fats that are taken up into the liver are actually burned by enhanced mitochondrial activity, and that's why we have never observed any indication of fatty liver development or steatosis or so forth.
Thank you.
Go, Carolina, and then. We'll get to you.
Thank you so much. It was really very interesting talk. Carolina from Steno Diabetes Center. I'm very interested actually in this. You said that some of the cachexia conditions, they're given extra energy source. For example, in the super premature babies, they get a mix of amino acids because this is intravenously, because this is the easiest kind of to take into, so they don't have to process this. I mean, has ever been anything done in these conditions to actually show where is the problem, if it's actually the food that is not getting into the cells, so the basic compounds, or is it actually processing of the compounds that is the problem?
Yeah, you're addressing the million-dollar question in the field. I think there's a reason for the fact that there's not a single approved drug so far. And indeed, in the clinics, any extra caloric infusion, whatever, does not help. So I think the overall conclusion is, well, there is a certain degree of anorexia, so you take in a little bit less calories because patients don't feel that well. However, as you cannot compensate or counteract this by additional calories, this cannot be the only explanation. Second, there is probably in certain types of tumors, talking about pancreas, talking about colon, a certain degree of malabsorption, also in the intestine, right? So that you get in a little bit less calories. But again, how do you then explain cancer cachexia in lung tumors? So what I'm saying is we do know that in the periphery, and that holds true probably for adipose tissue, definitely probably for skeletal muscle, for the liver, there are obviously futile cycles going on that burn essentially energy whenever it comes in, and so it cannot be stored anymore. And what exactly these futile cycles are under these conditions is not yet clear. There's a big debate and ongoing research at the moment. But all three together basically explain a little bit why we think it's so difficult to treat patients and why it's so far not possible to actually counteract it, because there are at least three components that you have to tackle. So the overall conclusion for now for the field is that you probably have to have a multimodal approach, including probably, well, nutritional support. You have to counteract the futile cycling in the periphery. And you maybe want to have some exercise, actually. That seems to be beneficial for some patients to just keep muscle strength and also muscle quality. So all this together is probably giving this phenotype. And it's not clear yet whether the phenotype is identical between different tumor entities, so whether lung cancer is really identical to pancreatic cancer or colon cancer. And what are the mediators that drive these whole things? We have identified, I have not talked about this, we know, of course, that the tumor secretes certain, what we call them cachexokines, that drive cachexia phenotypes in the periphery. And some of them, if you basically block them then in the circulation, you are able to at least partially prevent some of the cachexia that you see. So it's still a big thing and not clear yet for every type of tumor. Maybe one final thing I say, because I have adipose tissue people here, it is not UCP-mediated thermogenesis.
Don't look at me.
That is important for cachexia, because there have been some high-profile papers in the past, but by now it has been real clear by using UCP knockout mice, etc., that this is not affecting cancer cachexia, and in particular not in humans.
Thank you so much.
Excellent. Well, the microphone's going up there. So when I saw the no, no argument there. With the combo mirror, have you, when you mentioned this thing about mitochondrial biogenesis, then my immediate thought is, have you ever paired the combo mirror with the liver-specific uncouplers, which can lower weight, so maybe you can get sort of everything in one?
No, haven't done that yet.
You should.
Good idea, but no, haven't done that.
Hi, thank you. It was a very nice talk. So I see that you see a lot of benefits from fasting that you say are dependent on the fact you don't have nutrients, that you see upregulation of these different factors. And I'm wondering what your opinion is on the fasting mimicking diets, and if this is actually just no nutrients, or maybe there's actually specific nutrients that are inhibiting some of these positive factors.
Mm-hmm. So the clinical study that I showed you was actually done with fasting mimicking diet. So these patients received this diet, which is a kind of a low caloric ketogenic, diet, and it was compared to a Mediterranean diet, and it had these benefits in terms of diabetic nephropathy. I do think you can mimic the same. You don't necessarily need this fasting mimicking diet, but there's this famous 18 to 6, fast 18 hours, do time-restricted eating then in 6 hours. And so this also has been shown to have beneficial effects, both in mice and also in clinical studies. And it works also in diabetes or in healthy people. So I think the main assumption is that whatever you do, you have to basically give the pancreas a break. So reducing the frequency of insulin secretion over the day, I think that seems to be one thing. And then to induce, let's say, autophagy, I think that's the other second thing that happens under these conditions. And whether you achieve this by low calories or by really fasting for a certain amount per day doesn't matter so much. I do think the important message is that if you want to have, and that's what we know also from the clinical studies, in order to maintain the benefits, you basically have to do this constantly. So once you get off either the fasting protocols or the fasting mimicking diet, then metabolic, let's say, problems will reoccur after a certain time. So it's not that you maintain a healthy state throughout your life. And that's why I think it's just important that you do it in a way that you can build into your daily life without losing quality of life. And if you skip breakfast and then just start eating at lunchtime and then have a normal dinner, that works for many people, as we know also from clinical studies, and that has benefits. And you can do this, and if you don't do it one day, then it's not so bad, but you can restart it easily. So I think the duration of these signals is important to achieve the metabolic benefits. Maybe that's the answer.
Make a very short question.
Okay. Thank you so much. It's a very beautiful talk. I'm personally doing IF, so that's also a very interesting topic to me. So I was fascinated by the interactions between cell types in liver. So if I'm not wrong, I think probably the second biggest or the third biggest population in liver is endothelial cells. And we do know in a lot of physiological conditions, there are like known interactions between endothelial cells and liver cells. And do you think there will be like any fasting-dependent interactions between those cells? Have you considered probably performing single-cell omics experiments to investigate this matter, like in an unbiased manner?
Yeah, so the simple answer is yes, we do have considered this, but we simply haven't done it yet. But my prediction would be, from all we know, that of course there's so many crosstalk going on between different cell types that the macrophage hepatocyte was just one little example of things that might have to come together to mount the full fasting response. I would be sure that endothelial cells, whatever stellate cells, I mean, you name it, they probably all contribute in one or the other way to certain degrees. In the end, it's maybe the hepatocyte, if we talk about metabolism, that then produces the ketone bodies or so and the glucose, right? But the others will have stimulatory impact on this, but we simply haven't done it, but it's a great thing and probably many things still to do in the future. Yeah, thank you for the comment.
Excellent. Thank you again, Stephan.
Thank you.